Okay, before we get into 7.3, let's do a review question. So, in the following diagram, NAD+, this molecule on the left-hand side, is A, gains electrons and is oxidized, gains electrons and is reduced, loses electrons and is oxidized, or D, loses electrons and is reduced. So go ahead, take a look at this and select your answer. Pause the video if you need to. Okay, the answer would be B. It gains electrons, which means it is reduced. So this molecule here is gaining 2 electrons, as well as 2 hydrogen ions, and is becoming reduced through this process. Remember, gain of electrons is reduction. That means this is a higher energy state, and we're storing some energy that has been transferred to it. Okay, so we're going to see this process in the breakdown of glucose. So we're going to start first talking about taking glucose and breaking it down. This is called glycolysis, which means to break glucose. There are some options is what we can do with those breakdown products called pyruvate. We can send it to fermentation, which really doesn't produce much energy, or we can keep extracting energy from it through this TCA cycle and send it on to respiration, which is the next section. So we'll talk about how it goes from glucose into pyruvate. This is called glycolysis. In that process, we're going to make a little bit of ATP and some NADH. These are energy storage molecules that will be used to power other reactions. You can further break down pyruvate, this end product here. You can either ferment it or send it to the TCA cycle. And then we'll talk about a little bit about how we use this in clinical labs to identify different pathogens. Okay, so we're talking about the breakdown of glucose, which is a sugar. It has 6 carbons. That isn't super important, but it is kind of, because each carbon-carbon bond is stored energy, and they're going to get broken and release energy, which will be captured in different ways and stored in different ways. This overview image here is really important. This is a microbial cell, and this is the full pathway for the most efficient breakdown of glucose. There are other ways to break down glucose that yield less energy, but this is the way to extract the most amount of energy from it possible. Your mitochondria do a very similar process to this, so it is not unuseful to know this for microbes, because it does apply also to humans. So let's walk through this overview image. It's not going to make a lot of sense yet, but I will keep showing this image over and over again to keep reorienting ourselves. So there are many different sources of energy. The simplest one that feeds into this pathway that we're going to talk about is glucose, which is a 6-carbon sugar. The first step is to break up that glucose. You basically split it in half, and you generate 2 molecules of what are called pyruvate. In the process, you're going to make 2 ATP, and you're going to have some high energy electrons that get sent and stored in this NADH. This NADH is not going to do anything until much later in the process. That pyruvate can go be fermented, which kind of just finishes the process, doesn't really extract much energy from it. If you want to get more energy, you need to break the pyruvate into smaller molecules called acetyl-CoA, which get fed into this cycle called the Krebs cycle. You can see here, we're chopping carbons off, and in the process, releasing CO2. So if you think about your own body, right, you breathe in oxygen and you exhale CO2. This is where that CO2 is coming from this breakdown of pyruvate here. In the process, more electrons are transferred and stored, and a little bit of ATP is also made. Again, more electrons are stored. So overall, we have like 4 ATP that's been made. Not a lot, but we have all these electrons here. In the final part of energy production, aerobic respiration here, you're going to go through oxidative phosphorylation, where all of these electrons get sent to the membrane to this thing called the electron transport system. Its job is to pump hydrogen ions outside of the cell. They're going to form a high concentration out here, and then they're going to flow back through this enzyme called ATP synthase. This is where most of the ATP is generated. You can see 34 ATP is going to be made here, which compared to 4 is quite a lot more. So going all the way through to this oxidative phosphorylation is a big part about being efficient with your energy production. You need to do this process, right? You cannot survive on just glycolysis and fermentation. Some microbes, though, can survive off this. It's not very efficient, but it works for them. We are too large to do that, so we need to go all the way through. And in this process, this is where oxygen comes in. It helps pull this stuff through this system, through the electron transport chain. So we'll see each of these pieces in the various sections that follow. But first, we're going to start with glycolysis. So glucose is a sugar, and glycolysis is glycolysis. Lysis means to break, so breaking of glucose. It basically splits it in half, and some of that energy is going to go into ATP. This happens in 2 steps. In the first stage, we actually have to spend some energy. We actually have to consume some ATP. But in the second phase, we will release ATP. So net, you're going to get about 2. This is kind of like you have to spend money to make money, right? You gotta get this process going by spending a little energy. Okay, please do not memorize the components here, but I'm showing them because I want you to see what's happening. So here's the overall reaction. You have C6H12O6. That is glucose. Okay, 6 carbons linked together. We're going to split that in half, and we'll end up with 2 molecules of C3H4O3, basically half here. Some of the hydrogens are lost because they're sent to this energy storage molecule, NAD+, which becomes NADH, which has hydrogen ions and high energy electrons there. We're going to take ADP, add some phosphates, and make ATP in the process. To do this, we start with our glucose, and on the y-axis here is kind of the energy state. Glucose is at this energy state. You can see we're actually going to spend some energy using 2 ATP to bring it up to a higher energy state. And then it's going to come crashing down to a much lower energy state, and we're going to end up with 2 3-carbon pyruvates. In the process, NADH is made and ATP is made. So you could write this reaction: glucose plus 2 ATP as inputs become 2 pyruvate, 4 ATP, which is a net of 2, and 2 NADH molecules. Is it important to memorize this formula? No. Is it important to know that you're breaking a bond and extracting that energy? Absolutely. We're going to watch the animation for this. It's probably going to go in a little more detail than we need, but I like you to see it in motion.
Glucose can be completely broken down to yield energy through a few consecutive metabolic pathways, beginning with glycolysis. In glycolysis, glucose, which has 6 carbons, is split into 2 molecules of pyruvate, which have 3 carbons each. Overall, a small amount of energy is released, some of which is captured by energy-carrying molecules, NADH and ATP. Here the reactions of glycolysis will be arranged vertically based on the energy content of the substrate molecules. Glucose contains more energy than the 2 pyruvate molecules combined, which is why energy-carrying molecules can be generated during glycolysis. Glycolysis occurs in 2 stages. Stage 1 actually consumes energy, 2 ATP molecules, in order to prepare glucose for a controlled breakdown that ultimately releases more energy than is invested here. In stage 1, 2 hydroxyl groups on glucose are replaced with phosphoryl groups from 2 ATP molecules, and the molecule is slightly rearranged. The resulting molecule is called fructose 1,6-bisphosphate. This sugar then splits into 2 3-carbon molecules of glyceraldehyde 3-phosphate, each with a single phosphoryl group. these substrate molecules are primed to start releasing energy. In stage 2 of glycolysis, each of the two G3P molecules loses 2 electrons from 2 hydrogen atoms, converting NAD+ to NADH. NADH is a reducing energy carrier. The oxidation of G3P releases enough energy to add inorganic phosphate to each molecule, forming 2 molecules of 1,3-bisphosphoglycerate. Subsequent reactions transfer both phosphoryl groups from each molecule to a total of 4 ADP molecules, making 4 ATP molecules. Notice that although 2 ATP molecules were consumed in stage 1, 4 ATP molecules are produced in stage 2, for a net generation of 2 ATP molecules from glycolysis. At the end of glycolysis, the remaining 3-carbon molecule, pyruvate, no longer carries phosphoryl groups, but still contains a significant amount of energy that can be harvested by further oxidation.
Okay, we're going to pause there because the animation is going to keep going, but we have to talk about what's going to happen now. Okay, so glycolysis has given us, right, some ATP, released some of the energy, and we have 2 molecules of pyruvate here. This pyruvate still has energy because it still has 3 carbons bonded together. You need to complete the breakdown of this, and you have 2 options to do this. One is fermentation, and this is not very efficient. It kind of, how do you say, I guess it short circuits and just kind of extracts a bit of energy from it, just a tiny bit, but really doesn't use it. It just kind of gets rid of the byproducts here. The other option is to keep extracting energy from the pyruvate molecules through a process called respiration. We're going to look at fermentation first, and then we'll look at respiration. So in fermentation, that NADH that was generated is going to return some of those electrons back to pyruvate. So we're actually giving back some of the energy we gained. And basically, it forms organic acids or alcohols as a product. It doesn't use any oxygen, and really it gets rid of byproducts is kind of the process of this, but basically no energy is made in this process. So you only get that energy, those 2 ATP from glycolysis, and this just deals with the leftovers from it. Now, fermentation works well for microbes because they don't have huge energy needs like we do. So things like bacteria and yeasts will often do fermentation. You're probably familiar with ethyl alcohol fermentation. So in this case, pyruvate, right, pyruvic acid here, is broken down. Some CO2 is released. That's why beer is fizzy and bread rises. But the NADH is basically released, and those electrons are given back, and ethanol is made in the process. So Saccharomyces cerevisiae does this, baker's yeast. There are other pathways. E. coli can do some where it makes succinic acid. It makes something else. You might be familiar also with things like yogurt, cheese, butter. Those often have lactic acid in there. So there are bacteria that do lactic acid fermentation. And you can actually do some lactic acid fermentation. When you go into anaerobic workouts, you start to produce lactic acid because you can do fermentation. It's like a last-ditch resort, but you can't do it for very long because you don't make enough energy through it. There are other things that are important, making other alcohols like isopropanol, butanol. These are very important. There are some certain types of propionic acids and alcohols that are made by Swiss cheese making bacteria that give it its specific taste of Swiss cheese. So there's all kinds of different things that can be made by different types of fermentation. And we have uses for these, right? These are industrial solvents and chemicals. Of course, we eat these things, alcohol and bread, baking. So these processes are not giving any energy, but they do produce some products that we can use. And really, for microbes, these are waste products. Ethanol, lactic acid, they can eventually build up and prevent the growth of microbes, both other species and the ones that create them, right? Yeast can only make so much alcohol until they can't survive in that. That's why beers and wines do not go up to a super high percentage of alcohol. You actually have to do distillation to get higher percentages there. This is not efficient as respiration, right? So this is just like, hey, I'm just going to get rid of this stuff kind of mentality. For us, fermentation is awesome for identifying microbes because different microbes do different pathways there. In the case of pathogenic E. coli, there's a test called the sorbitol fermentation test. So we can see if pathogenic E. coli ferment sorbitol or don't. And in this case, non-pathogenic E. coli will ferment sorbitol and will make these red or pink colonies, whereas pathogenic E. coli, ones that would cause like bloody diarrhea or things like that, they don't ferment sorbitol. They've lost that ability, and they end up producing white colonies on here. So if we had someone with gastrointestinal issues and we thought it was an E. coli issue, we would plate it out and look for these white colonies because they would indicate pathogenic forms of E. coli there. We've seen fermentation for other things, mannitol fermentation for Staph aureus versus Staph epidermidis. We've seen other E. coli lactose fermenters, things like that. So fermentation is huge in terms of being able to identify what's going on. And a lot of that has to do with using the byproducts of fermentation, which are acids that will change the pH of indicator dyes. So fermentation is one option. It's not the most energy efficient one, but it works for some microbes. If you want to extract more energy from that pyruvate, you can do respiration, where we keep removing electrons from pyruvate to make more NADH. NADH carries both electrons and hydrogen atoms ultimately to that membrane-associated electron transport chain, and it's like a carrier molecule. It's taking some of that energy and moving it, and that's going to basically be used to generate what we call the proton motive force, which will produce lots of ATP. This might involve oxygen at the end, right? That would be aerobic respiration. Sometimes you can have non-oxygen final electron acceptors. That would be anaerobic. Aerobic is the most efficient. It produces the most energy. Anaerobic produces less energy than aerobic, but still a lot more than fermentation. Okay, so we've broken down our glucose into 2-pyruvate. We could just kind of short circuit and finish up with fermentation, or we can shove it into respiration. For respiration, these 2-pyruvates are going to get broken up into 2-acetyl-CoAs, and then those are going to get sent into this TCA cycle that we'll see here in just a sec. So the TCA stands for tricarboxylic acid cycle. And really what this is doing is taking the pyruvate and transferring all the electrons that are in there to these shuttle molecules, NADH and FADH2. In the process, a little bit of ATP is made, but not very much. So don't, again, memorize this, but we start with a 3-carbon pyruvate, and we chop one of those carbons off to make a 2-carbon acetyl-CoA. That carbon gets lost as CO2. In the process, we steal some electrons there. We steal their energy. This is where other pathways can meet up. So lipids, amino acids, they can all kind of jump into this cycle at this point. This 2-carbon molecule is going to go into this cycle, and carbons are going to get cut off of it. And that's going to release CO2 and produce some energy in these electrons here. This just keeps coming down and going through, and the end product is some stored energy and CO2 molecules. This process is also called the Krebs cycle because of the person who discovered it, and the citric acid cycle because there's citrate involved. So if you see those names, don't be confused. Basically what this is doing is just chopping this molecule up until it's just CO2 and extracting the energy from it. Again, though, we've only made a small amount of ATP. ATP is our most stable storage molecule for these these high energy electrons. We do have the carrier molecules, NADH and related FADH too, but we're going to use these that we've produced to make a lot of ATP later when we get to the electron transport chain in the second part of respiration. Remember, this next part can either involve oxygen, that's aerobic respiration, or not involve oxygen, anaerobic respiration. Okay, let's watch the rest of that animation on the TCA cycle.
Pyruvate will be completely oxidized by two additional pathways. In the first, catalyzed by the pyruvate dehydrogenase complex, pyruvate is stripped of a carbon, in the form of carbon dioxide, oxidized by NAD+, forming the energy carrier NADH, and combined with a molecule called coenzyme A, forming acetyl-CoA. In the next pathway, called the TCA cycle, acetyl-CoA combines with a 4-carbon molecule called oxaloacetate to generate a 6-carbon molecule called citrate, or citric acid. Citrate is a tricarboxylate, a triple acid, and gives the tricarboxylic acid cycle its name. This pathway is also known as the citric acid cycle, or Krebs cycle. In the TCA cycle, the two remaining carbons are removed in the form of 2 carbon dioxide molecules. The energy released by the removal is captured in energy carriers, ATP, FADH2, and NADH. As a result of these reactions, the 4-carbon molecule, oxaloacetate, is reformed and can be used in another round of the TCA cycle. From these catabolic pathways, glycolysis, the oxidation of pyruvate, and the TCA cycle, a small number of ATP molecules are produced. However, NADH and FADH2 are also formed, and they will transfer their high-energy electrons to the electron transport system, or ETS. In the ETS, the energy is transformed, ultimately producing a large number of ATP molecules.
All right, so again, I want to reiterate that the differences in metabolism between microbes, it's really important to identify different pathogens. So there are a lot of quite rapid tests, actually, to identify different microbes. They do rely a little bit on growth, but these microbes produce these fermentation products quite quickly. So in this case study, we have Mark. He's born prematurely at 30 weeks, weighs 1.7 kilos or 3.75 pounds. So that's quite small. And his mother was a 15-year-old woman who didn't have prenatal care. So we have some issues going in here. It's not the ideal situation. Seems fine when he's born, but within a few hours, starting to have respiratory distress and is intubated and has mechanical ventilation, so assisted breathing. We're going to look for any sort of bacterial infections because we want to deal with those very quickly because it's a newborn, don't have a very robust immune system yet. And so we're going to take a blood culture, and at this point, there's no bacterial growth in it, but we do suspect some sort of problem. So we're going to do a little preemptive care. We suspect there might be issues, and to protect them, we're going to prescribe ampicillin and gentamicin, which are two antibiotics. By day 5, he's showing some problems, right? He has metabolic acidosis, so his blood pH is low, thrombopenia, low platelet count in the blood, and his cerebrospinal fluid has white and red blood cells in it, which is not good, right? We've talked about cerebrospinal fluid several times now. It should be clear and not have any of these things in it. So we're going to culture that, and it's going to grow a gram-negative rod. And they can find that now in his blood. So he has some sort of bacterial infection. Because he's so small, it could be quite serious because it can circulate throughout the body. So we need to identify immediately what the organism is in there. So we're going to do some diagnostic tests. In this case, they're going to use Simmons citrate agar, which has a color change that occurs with it. And they're going to do another one called phenol red tests for fermentation of dulcitol and Malebios. Sucrose is negative, so they're running a bunch of different fermentation tests. They're going to test for arginine dihydrolase, which is an enzyme, Ornithine decarboxylase, another enzyme, those are positive. Lysine decarboxylase is negative. So we have what? 1, 2, 3, 4, 5, 6, 7 different tests here that are all kind of relying on metabolism. Okay, do I want you to know anything about any of those tests, right? No, not really. Obviously, those are complicated processes, and if you're a clinical microbiologist, yes, you would need to know about them. What it does tell us, though, is we can get a pattern from that. Certain organisms do certain things, and the more data points we have, basically it becomes like a fingerprint. We can closely identify what might be causing this just by those tests, which can be done quite quickly. And in this case, they're going to identify it as Citrobacter in his blood, which is causing septicemia and meningitis, which is pretty rare. But they're going to take that bacteria and test it with different antibiotics to find which antibiotic works best for it. And in this case, after 10 weeks, that antibiotic therapy is going to probably save his life, and he's going to make a full recovery. So this testing of cultures for different antibiotics they're susceptible to or can be killed by, that is something we're actually going to do in our own lab. So you will get some experience with that. But that's a huge, huge thing, right? Because different strains of the same species can be resistant to certain antibiotics. So we got to test that. Another kind of case study here is a way that metabolism can help us environmentally. So as we all know, oil can and generally will spill at some point, and when it gets into marine environments, that really is difficult to deal with because the oil stays on top. It really can screw with ecosystems. It can cover animals and things like that. Luckily, it doesn't stay there forever because there are certain marine bacteria that have enzymes that degrade the compounds found in crude oil. They've evolved to do this because slowly oils and things seep out of the ground naturally. So it's kind of a slow process, but when there is an oil spill, there are thoughts that maybe we could bioengineer these microbes to help degrade the oil spill. They take what are called aromatic compounds. They have these carbon rings, and they break them down into acetyl-CoA, and then they shuffle that into that TCA cycle we talked about. So this isn't something we can do, or in fact most bacteria can do, but there are certain microbes that will do this, and they send it to the TCA cycle. So this is like their food, right? Oil is their food because it's an organic molecule. Ultimately, this will degrade the oil in the environment, but depending on how much is released, that might take quite a while. All right, 7.3, we talked a lot about glycolysis, taking that 6-carbon sugar, breaking it into 2 3-carbon ones. Little bit of energy is stored along the way. Some microbes go into fermentation, but really don't make much energy from that. They just make waste byproducts. You could also go into respiration. That pyruvate gets into acetyl-CoA, which goes into the TCA cycle. That just keeps chopping carbons off and releasing energy, storing that as a little bit of ATP, some NADH and FADH2. In the next section, we'll see what happens with these. In the process, because we're chopping carbons off, CO2 is made. So that's why a byproduct of our respiration is CO2. These fermentation pathways are very critical to identifying specific microbes, and doing lots of different tests will tell us quite precisely what species the microbe is. Okay, that's it for 7.3.